Hey Dr. Crumb, here is my video answer. So you have asked us to illustrate the phases of mitosis. So I will start with late G2. So in late G2, the cell has already copied its DNA. So it's going to have two copies of DNA. Boom, boom. And because the chromosomes are not completely condensed yet, they're not very clear and they're still long and stringy. Also, the cell has copies of its centrosomes. So it has two copies of centrosomes. And then the next phase is early prophase. So with early prophase, cells start to break down some structures, but they also start to build others up. So chromosomes start to sort of condense to where you can see them. They're not as stringy anymore. So I'll draw those. Trying to keep with the same colors. And also, a mitotic spindle starts to be created between the centrosomes. Oh, I'm not using the same colors. There's the mitotic spindle. So the mitotic spindle is made of microtubules that organize the chromosomes and move them around during mitosis. And then we have, this is not pretty at all, late prophase. And with late prophase, chromosomes completely finish condensing. So you'll have your condensed chromosomes that you can see clearly. And your centrosomes move to opposite ends. Also, the nuclear envelope has completely broken down, so that's why the chromosomes are released. And the mitotic spindle starts to grow and it'll attach at the kinetic cores. So the kinetic cores are these little center parts of the chromosomes where the microtubules attach, and the purpose of them is to give an attachment site to where whenever the chromosomes are pulled apart, they are pulled apart evenly and distributed evenly. So then we have metaphase. So with metaphase, chromosomes are completely lined up in the middle. Now, this is not like an actual physical site, but it is called the metaphase plate, where they're lined up completely in the middle and they are ready to divide. So our centromeres, mitotic spindles are attached to the kinetochores. So there are two kinetochores on each side of the chromosome. It's really hard to see right now. That attach to microtubules from the opposite spindle poles, which are these spindle poles. Cells check to make sure all chromosomes are at the metaphase plate with kinetochores correctly attached to microtubules. So the cell takes a second to make sure everything is correct, and that is called the spindle checkpoint. And that is to ensure that sister chromatids split evenly, because if not, then lots of homeostasis stuff would not go right. The two sisters would not be the same. So then we have anaphase. So with anaphase, the chromosomes start to be pulled apart. So we'll have the chromosomes pulled apart. By the poles. Trying to make this color coded. At the kinetochores. So they're pulled apart from each other towards opposite ends, like completely opposite ends by the poles. And this process where they're pulled apart is driven by motor proteins, and these motor proteins will walk along the microtubule tracts and carry the cargo, which is the chromosomes. And the really cool thing is there are also mitotic spindles that are not attached to the kinetochores. And these mitotic spindles will actually push. So these that are attached to the kinetochores, attached to the chromosomes, will pull and be driven by motor proteins, but these mitotic spindles will push to give an extra force for separation. So after anaphase, we have telophase. It looks like a peanut to me, where the nuclear envelope starts, not completely, but starts to reform. So the cell is nearly done dividing, and it starts to reestablish like normal structures. Here are the chromosomes that are separated. Centrosomes are on opposite sides. So the mitotic spindle is broken down into its building blocks. Two new nuclei form, one for each set of chromosomes. The nuclear membranes and the nuclei reappear, and chromosomes begin to decondense back to their stringy form. So they won't be as easy to see. And then lastly, we have early G1. Early G1. So here, cytokinesis is complete, which starts right here. It starts to separate the cytoplasm. And there are two identical daughter cells with their little nucleolus and their new stringy decondensed chromosomes. So you may ask, like, what's the purpose of this? So mitosis is a type of cell division in which one cell, the mother cell, divides to produce two daughter cells that are genetically identical to itself. The cell in the context of cell cycle, my goodness, mitosis is the part of division process in which DNA of the cell's nucleus is split into two equal sets of chromosomes. So the mother cell and two equal sets of chromosomes in the daughter cell. A great majority of cell divisions that happen in your body involve mitosis. This is a very important part of any part of your body. During development and growth, mitosis populates an organism's body with cells, and throughout an organism's life, it replaces old, worn-out cells with new ones. So basically the goal of mitosis is to make sure that daughter cells get a perfect full set of chromosomes. Cells with too few or too many chromosomes usually don't function well. So that's why you have spindle checkpoints to make sure that they separate completely evenly. So you have two identical daughter cells, because if not, they will not function. They might not even survive, and they may even cause cancer. So when they undergo mitosis, they don't just divide their DNA at random. It's very specific. It doesn't just toss into piles for two daughter cells. It's very specific. They split up their duplicated chromosomes in a very careful and organized series of steps.